Welcome everyone. Today we will complete our whole of Unit 2. As you know, we have already completed Unit 2. So today we'll just revise this part because this has some parts which are very important and interesting in research methodology. So, as many of you who are new and have come on to this video, let me give a brief about this video descriptions. We are doing 5 units of research methodology. In this, we have completed Unit 1 and Unit 2. If you want to see the previous videos, you can go to the description. Over there, you will get the playlist, and from there, you can watch the previous videos. If you want the PDF of these classes, you can go to the Telegram channel, Impractica Classe. Over there, I will provide you with the PDFs, and you can download it from there. So, let's start our video. So, when you are deciding on a research problem on which you will be doing a research, the first thing that you have to notice that what is your broad area of topic. Suppose you are doing your PhD on environment. You are completing your PhD on what? So, you are completing your PhD on environment. So, in that you have selected a broad area biodiversity. So, from biodiversity, you have to quantify or select the things which give you the most interest or the things which are most important, crucial at that particular point of time. Like biodiversity can be done both on land and both on water. If you select on land, on what you will do, on what part you will do, you can do your biodiversity research on animals, insects, bird. Suppose you select animals, so which animal will you select? You have selected tigers for your research purpose. So this is the way in which you have to narrow down and select your area of research topic. Second is we have to review the existing literature. The second part is we have to review the existing literature. So when you will do your PhD, you will read a number of articles. In each of the articles at the end, you will see a thing like this, which is called limitations. So you have to go on to the limitations and study which are the areas that further research can be done upon. Like there is a topic, suppose tigers population is decreasing in Madhya Pradesh. So you can do further research on that. They have said that tigers population decreasing due to what? Due to deforestation. You can do a research that tigers population is affected by hunting. How the tigers population is affected by hunting. You can select different research areas and the places for your topic. Like you can select a different state like Chhattisgarh. Your earlier article was done on Madhya Pradesh, so you can choose a different state like Chhattisgarh, or you can totally go for a different country, like you selected the country of China for your research. So this is known as reviewing existing literature and the research gaps which will help you to do further research. Now we move on to consider practical problems. So you have to consider the things which are the most relevant and which are the most current topics at that present moment. For example, in the year of 2021 and 2022, there were many research papers on the problem of corona, because at that time corona was all throughout the world and everyone wanted to know more about corona. So many research articles were done which were concerned about the corona. So you have to develop articles which are relevant to that particular point of time. For the sake of our example, we can understand that you have researched tigers population. You have seen in newspapers and articles that the tigers population is decreasing. So to make out the census of how many tigers are there, you have developed a mechanism of M stripes. M stripes was actually developed in the year 2010, but for the sake of example and for the sake of our understanding, we can say that a new mechanism has been developed to understand the population of tigers. It is M stripes, and you want to do research on that. So you want to find out the census of how many tigers are there every 5 years. So these are the new things, the practical problems you have to do your research on. Then we go on to formulate a problem statement. When you are formulating a problem statement, you have to keep three things in mind. The first one will be you have to describe the issue being addressed. So what is the issue we are addressing? So the issue that we are addressing in this particular research topic is the decreasing population of the tigers. Second, we have to see the significance of the problem. Why are we doing research on this topic? We are doing research so that to maintain the biodiversity of that particular place, because if tigers population goes down, the biodiversity in that particular area will also go down. Third is outline the scope of research. By the scope of research, we mean you have to say the particular area or particular domain or particular it is easier to remember that we have to describe the particular area or population in which your research will be limited to. For example, for the outline of the scope of the research, but we have decided that our place will be Madhya Pradesh. So based on these three, you have to create a title. See, the declining tiger population in Madhya Pradesh forest, impact on biodiversity. So this will create your problem statement of the title. The declining tiger population in tigers population, you can see this will be our first point. We have described the issue. Biodiversity will be our second point, which will show the significance, and our area of study will be Madhya Pradesh. So this will be how we formulate a problem statement. Next, we go on to the objectives. So in objectives, you have to mainly remember SMART. It is a short form. SMART is a short form. S stands for specific. You have to be very specific what you are doing. So in this, we are measuring the population of tigers. So second one is your measurable data. You need to have measurable data to continue with this research. For example, you have measured that in 2024 the tigers population is 90, and in 2023 the tigers population was 100. So the tigers population has decreased by 10. So it is a measurable thing. Third, we have to see achievable. A stands for achievable. Most of the PhD degrees we get a 5 years time. So you have to see that your research has been completed within 5 years. So for that, you cannot take the whole of country of India. You have to choose a particular state. In this case, we have chosen the particular state of Madhya Pradesh for doing our research. Then we come on to R. R stands for relevant. Relevant, as we have all discussed about, that your topic should be relevant. Like national parks and AIMS tribes, all this, your topic should be relevant. And D is for time bound. You need to have idea that when you will set this and complete this project, how much time you need to set your title, how much time do you need for your literature review, how much time do you need for your data collection. All of this should be set up, and your complete research should be completed in a time bound manner. Then we go on to align the objectives with your research problem and question. So your objective should be placed in such a way that they answer to your title. For example, here we cannot say we want to identify the biodiversity in Madhya Pradesh. We are not dealing with the biodiversity of whole of Madhya Pradesh, but we are only dealing with the biodiversity in relation to tigers. We are dealing with only to the relation with tigers, how the biodiversity of the particular place, in this case Madhya Pradesh, is affected. So your objectives have to align with your research problem. Third point is you have to use action verbs. Action verbs are what? You have to use to identify, to describe, to analyze, to evaluate. So you have to use verbs which will say that you are taking some actions. Like you can say to identify the reason for the decreasing tigers population in Madhya Pradesh, to describe the criteria of the biodiversity in Madhya Pradesh jungles. So all these things you can use, and use always action verbs in the objectives. Then we will see the same things. Make objectives specific and measurable. Objectives should be achievable and realistic. Make objectives relevant and significant to that particular topic. And you have to structure your objectives logically. Like for in the first one, you cannot, you have to structure your objectives logically. So let's go on to the previous part to understand this. So we have taken the topic, the declining tiger population in Madhya Pradesh forest, impact on biodiversity. So first we have to see what is the first point, tigers population. So first objective we will say to study on the reason for the decreasing population of the tigers. So this will be our first objective. So we have to arrange our objective in a logical sequential manner. Second, we can say to analyze the impact on the state of Madhya Pradesh with the decreasing population of tigers. See, second point we have included Madhya Pradesh. So in your objective also you will include according to the sequence in which we have given the title. Now we will move on to hypothesis. So this is where the interesting things start. So before understanding hypothesis, we have to understand what are variables. Now variables. Variables can be any factors which are used in our research. So variables can be anything like air, water. These are all called variables. Now variables are mainly of two types. One is called independent variable and another one is called the dependent variable. Independent variable is controlled by the researcher and their effect is shown on dependent variable. That's why independent variable is known as controlled variable and dependent variable is known as the affected variable. For example, we will do a research on drinking tea as effect on sleep or not. So through research, we come to know that the more amount of tea you drink, the lesser will be your amount of sleep. So in this, the independent variable will be the tea and the dependent variable will be the sleep, because we can change the amount of tea that we are giving to the particular person, and the person's effect on sleep, the person's effect we will measure. So sleep will be our dependent variable. And how will be your hypothesis? Your hypothesis will be a combination of both of these two factors. Like your hypothesis will be the person who drinks tea sleeps less than a person who doesn't drink tea. So this will be your hypothesis. Now, hypothesis is also of two types. One is called alternative hypothesis and the other one is called null hypothesis. Your null hypothesis will always show that there is no change, and alternative hypothesis will show that there is change. Alternative hypothesis is represented by H1 and null hypothesis is represented by H0. So alternative hypothesis will show that the person who drinks tea has less sleep than a person who doesn't drink tea. In the same way, the null hypothesis will show the relation that tea and sleep doesn't have any relation. I hope you have understood this. Let's understand this with another example. So in this example, we have taken that the more a student studies, the more will be the marks. So study will become our independent variable and marks will be our dependent variable. So alternative hypothesis will be what? It will show the change. More study will be equal to more marks. And null hypothesis will show us that there is no change. So it will show study has no relation with marks. So this is null hypothesis, and second one is the first one is the alternative hypothesis, and second one is the null hypothesis. So what is the role of a hypothesis? What is the use of making hypothesis? The hypothesis provides us the direction and focus. So by hypothesis, we know that in what way we have to do that particular research. It guides the research process. It defines the variables. Like variables are what? Over here in our first study, the variables are study and marks. In second one, our variable is sleep and tea. So these are our variables, which defines our variables. Fourth one is facilitates testability. So we have to test these variables and see if the null hypothesis is true or alternative hypothesis is true. It enables structured analysis and interpretation. So after we have conducted the test, then only we can say, interpret the results and say that whether they have effect or do not have effect on each other, and they contribute to theory development. In the first one, we have developed the theory that more a student studies, the more will be the marks. So this is called theory. In second one, we have developed a theory that a person who drinks more tea has less amount of sleep. So these are called theory development, and these are the basis of our statistical testing. Now we will move on to the formulation of hypothesis. So we have already studied how to formulate a hypothesis. In this, we will go on to a little more detail. So we have to conduct and identify the research question. What is the research question? In the first case, the research question is how much amount of tea has the effect on sleep. What kind of effect tea has on sleep? So this is our research question. Then we have to conduct a literature review. We have to see the literature, if anyone has done the same kind of research or not, what kind of study has been done on tea. All of this thing we have to do in literature review. Third, we have to identify the variables like tea, sleep, study, marks. All of these are variables. Now the important part is we have to define the relationship. So relationship can be of two types. There can be a negative relationship and one can be a positive relationship. In negative relationship, we can see the more amount of tea we have, the lesser amount of sleep we have. So this will be a negative relationship. That if one factor increases, one variable increases, the other variable will decrease. And in positive relationship, what we will see, that more amount of study we do, the more amount of study we'll do, the more amount of marks we will get. So this is a positive relationship because both the things are moving in the same direction. The more amount of study, more amount of marks. Now, direction will be of two types. After doing the test can have two results. It will be alternative hypothesis, which will show a change, or it will be null hypothesis, which will show there is no change. Now, alternative hypothesis will be of two types. It can be of one-tailed or it can be two-tailed. One-tailed, it will say that there is definitely a change and it will show a clear direction in which the change is applied. Will show a clear direction in which the change is applied. Example, study will show that more marks we can get. Study will show the more marks we can get. Suppose it is said that a person who drinks 4 cups of tea will sleep for 2 hours of less. So in this we can understand the direction is given in each of these examples. So these are called one-tailed test. In two-tailed test, what it will be told, that a change is there but we cannot set up the direction. We don't know whether it will be a positive direction or it will be a negative direction. Over C, you over here we can say that study will have a marks on effect, but we don't know if it will have a positive effect. Sorry, it will have a positive effect or it will have a negative effect. That will be a no direction. In the second case, we can see T will have a effect on the sleep, but we don't know if it will have a positive effect or it will have a negative effect. These are called two-tail test. That change will be set, but the direction is not set. Now we will move on to the types of error in hypothesis testing. So first, before understanding these types of error, we have to understand that there are two types. One is null hypothesis and another one is alternative hypothesis. Now if null hypothesis is true, it automatically means that alternative hypothesis is false. And if null hypothesis is false, it will automatically mean that alternative hypothesis is true. I hope this part is clear to all of you. Now we will move on to testing. While we test, we can have two results. We can have null hypothesis is true. When I say null hypothesis is true, it means that null hypothesis is correct. In the terms of research, it is called null hypothesis true. And we can say that null hypothesis is false. Can get one of the two results. Now you can see that these are the types of errors. So when it is error, what will happen? We will say null hypothesis is rejected even when it is true. That is why it is called error. Error is what? When we have created some mistake. So in first one you can see that null hypothesis is true, even then we have rejected it. And in type 2 we can see null hypothesis is false, even then we have accepted it. When it is false, when it is wrong, even then when we have accepted it, it is called error. And when null hypothesis is false, what will happen? Automatically alternative hypothesis will be accepted. And in this case, when null hypothesis is accepted, what will happen? The alternative hypothesis is rejected. So this is called type 1 error, the first one, and the second one is called type 2 error. I hope up to here everything is clear. Then if we see onto it, we are saying this is true. So we will say this is positive. And over here we are saying this is false. Null hypothesis is false. So we will say negative. And because it is error, so we will add on the term false and over here false. So it will be easier for you to remember. So remember, type 1 error is called false positive and type 2 error is called false negative. Then type 1 error is also known as alpha error and type 2 error is known as beta error. So I hope everything is clear up to here. It can be a little confusing for those who are understanding it for the first time. Please go through this video once or twice and I will also provide the PDF. The PDFs will definitely help you to make this concept even more clear. So this is type 1 error and type 2 error. Now we will go on to the probability value. So probability value is very easy to understand. Probability value is the value of randomness. That how random can a result be. Suppose you have done your test whether the T affects the people sleep or not. For this you have made two groups of 10 people each. I hope this is clear. So for that experiment you have created two groups in which both the groups they are 10 people each. For the first 10 people you have not given them tea. Have given them any other drink which is not tea and you have measured how much amount of sleep they get. So you see that they get 8 hours of sleep. For the next group you have given them tea and you have measured how much amount of sleep they get. So over here you have seen that they get 6 hours of sleep. From this you can understand that if you give a person tea, the sleep is reduced. But now the probability value, or in research as we know it, the p value comes into place. Now these 10 people that you have selected randomly can have different these 10 people can have different sleeping patterns, or their genetic constitution can be different for which they sleep less, or they can have a habit for which they can sleep less. So all these random factors can be there in your experiment. So to accept all these random factors, a p-value is taken. A p-value is taken. P-value is what? It only describes that the probability or your randomness in your particular experiment. So if your p-value is more, what does it mean? That there is a high chance that the random persons will have these things, and your result is also a random thing, and your evidence will not be strong or accepted. And if your probability value or your randomness is low, then what? It will be accepted that your test has strong evidence and your test will be accepted. Your test will have a low random chance. So this is p value. Now we will move on to the significance value. This is the last topic of our chapter, significance value. What will significance value show? What will significance value show? In the previous slides, we have studied about type 1 error. I hope all of you remember type 1 error. So type 1 error is nothing, significance value is nothing, but it is equal to the type 1 error. So whatever the chances of having type 1 error is known as alpha, or the significance level. For example, we have taken that our alpha value will be 0.05%. When we say our alpha value is 0.05%, what do we mean by that? That there is a 5% chance that the experiment that we are doing will have type 1 error. There is a 5% chance the experiment or the research we are doing will have a type 1 error. Or we can also say that there is a 5% chance that the null hypothesis which shows that there is no change is true, but we are not accepting it. We are not accepting it. That is shown by the alpha level or the significance level in our research. Now, significance level will have two types. First one is our p-value, probability value, or our chance of randomness is less than the significance value. And second, p-value or the chance of randomness is more than the significance level. If our chance of randomness is less than the significance level, then our alternative hypothesis is true, which means that there is a significant change. And if our chance of randomness, our p-value, is more than the significance level, then our null hypothesis is true and there is no significant change that we can count. I hope the chapter is clear to all of you. Please go through this chapter once or twice and I will surely provide you with the PPTs, with the PDFs, which will be easier for you to open in your mobile and study. And if you have any questions, I encourage all of you to please post the questions. And if there are any changes, if there is anything positive or negative that you want to share, please do comment. I will be waiting for your comments eagerly. Thank you so much for watching this video, and we will meet in the next video, in which we will start with our Unit 3.